All right, guys, if you're following along with this playlist, we started off with an overview of cellular respiration and redox reactions. In the second video, I went into detail about glycolysis and fermentation. And in this third video, we're covering Krebs cycle, also called citric acid cycle or TCA cycle. And the idea is, if you picked up at the end of the last video, at glycolysis, we end with a couple of molecules of pyruvate. And remember, in glycolysis, we got some energy out of it, right? You made a little bit of NADH, we made some ATP directly, but these pyruvates still have a lot of energy in them left to be extracted, right? And so our next step in this process, if we have oxygen, right? My last video, we talked about what happens to pyruvate without oxygen. That would be fermentation. If you have O2, you're going to send that pyruvate into mitochondria, and you have to do that in the form of a molecule called acetyl-CoA. So there's a reaction that links glycolysis and the Krebs cycle together, and we call that reaction pyruvate oxidation. So for context, remember, glycolysis happens entirely in the cytoplasm or cytosol. That's over here. And this is showing the outer membrane of mitochondria. So we cross this line, we're entering mitochondria, and mitochondria have two membranes, an outer and an inner. And so this space in yellow, that would be the intermembrane space. This area in green, that's the very interior of mitochondria. That is the mitochondrial matrix. Okay, so we move our pyruvate through the intermitochondrial or intermembrane space. We enter the mitochondrial matrix, and that's where this reaction is happening. All right, so pay attention to the number of carbons we're working with here. Pyruvate is a 3-carbon molecule, and in the end, we're going to wind up with acetyl-CoA, which is only a 2-carbon molecule, right? So this reaction that's happening, which, by the way, they don't have it written in here, but the enzyme that's making this reaction happen is called pyruvate dehydrogenase, and big name, but it makes sense, right? Pyruvate dehydrogenase because we're removing these molecules, including a proton, hydrogen, from pyruvate, but you also remove a carbon. And so the fact that we're going from a 3-carbon pyruvate to a 2-carbon acetyl-CoA, that third carbon has to go somewhere, and that's our first CO2. So remember, our products of cellular respiration as a whole are CO2 and water. This is your first CO2 down. So keep track of these. We're going to lose the next 2 carbon dioxides as you go through Krebs cycle or citric acid. But for now, we're down 1 CO2. We add this coenzyme A, there's a cofactor, to that molecule, the now 2-carbon molecule, so we get acetyl-CoA. Right? This is the acetyl part. You tack the CoA on. It's eventually going to get removed and recycled. And that acetyl-CoA is what's going to enter Krebs cycle. Right? Along the way, we also oxidize pyruvate and handed off one of its electrons to make NADH. Okay, so we're entering mitochondria now with a couple things. We have 2 of these acetyl-CoAs, because remember, if we started with a glucose, we got 2 pyruvates. So we have 2 acetyl-CoAs per glucose, plus 2 of these NADH that are eventually going to take their electrons to the electron transport chain. All right, so this reaction shows it in quite a bit more detail. As always, it's kind of up to you to decide what level of detail your class requires. But this reaction up top shows us that same pyruvate oxidation that we just looked at. All right, here's your 3-carbon pyruvate. We're converting it into acetyl-CoA, and in the process, we get a little bit of NADH. All right, this citric acid cycle, which remember is the same exact thing as Krebs cycle, or you sometimes see it called the TCA, the tricarboxylic acid cycle, all the same thing. You're combining each acetyl-CoA with a molecule called oxaloacetate to produce citrate. So citrate is really the very beginning of Krebs cycle once you add your acetyl-CoA. And from there, this is very similar to glycolysis in that every step is just an enzymatic reaction. So aconitase and isocitrate dehydrogenase, these are all just different enzymes that oxidize our molecules to extract energy out of them, right? And pay attention, there's a lot going on here, but just pay attention to what your products are, right? So we've got a couple things to keep track of. One, we're making quite a bit of NADH right here and right here. We also, they don't have it written on this exact version of it, but at this step, we also make a product called FADH2, which is another electron carrier, right? More NADH up here. And in the process, you're eliminating CO2, right? So exactly what we want. We don't want to keep the carbon and the oxygen. The mass is not interesting for us. We throw it away, right? So you're just going to breathe out that CO2, but you've extracted energy along the way so that you're building all of this NADH, right? By the end of citric acid cycle, we've made 3 NADHs, an FADH2. We've eliminated a couple CO2. That's just a waste product. And we've also made 1 ATP along the line. Technically, you make GTP from this step, but that GTP is very easily converted into an ATP, right? We call it a cycle because by the time you get back around, you are right back to oxaloacetate again. And so we say it's a citric acid cycle because the whole thing regenerates itself, right? Your starting molecules, oxaloacetate and acetyl-CoA, enter the cycle. By the time you get back around, the carbons from acetyl-CoA have been eliminated as CO2. And when you finish, you are right back where you started. And that same oxaloacetate can now take another acetyl-CoA that came in from pyruvate oxidation and restart the cycle all over again. And so it keeps going around and around and around in a circle every time, take in an acetyl-CoA, spit off 2 CO2s, and make some NADH, FADH2, and ATP along the way, right? You probably noticed the amount of ATP that you're making is actually pretty trivial. It's really small. The bulk of our energy is coming from NADH and FADH2. They're going to take their electrons and hand them off to the next part of the pathway, which is the electron transport chain. And remember, for context, all of this is happening in your mitochondria. Okay, so back to our overview. All of those Krebs cycle or citric acid cycle reactions are happening in the mitochondrial matrix, right? This thing is a mitochondrion. Remember, it has 2 membranes, an outer membrane and an inner. This little space in here is your inner membrane space. Everything in white is the mitochondrial matrix. So these enzymes of Krebs or citric acid cycle are just floating around in that mitochondrial matrix, producing all of these products. The CO2 just diffuses out and makes it into your bloodstream to go to the lungs, and you breathe it out. And the NADH and FADH2 are being used to run the electron transport chain. And so what this little graphic is showing you is this electron transport chain, which is what we're looking at next. Those electron transport chain proteins are embedded in the inner membrane, right? So Krebs cycle enzymes are mostly just floating around in the matrix and the fluid of the mitochondria. Electron transport chain proteins are actually integral membrane proteins that are inserted in the inner membrane. And so, perfect. We're making this NADH and FADH2 right next to where the transport chain is running. And remember, we'll also get a little bit of NADH from glycolysis as well. All right, so in this view, pay attention to context a little bit. This is our inner mitochondrial membrane. They're not drawing it, but this would be our outer mitochondrial membrane out here. And so this is the intermembrane space, right? This is the mitochondrial matrix down below. All right, so try to picture a zoomed out view. We're looking inside of a mitochondria. Down here in the mitochondrial matrix, that's where Krebs cycle would be happening. Okay, and what we're doing is we're taking that NADH that we made from Krebs cycle and glycolysis, and NADH is just an electron carrier. So what is it doing? It is handing its electrons off to the electron transport chain. Right? These red proteins that you're seeing in the membrane, those are the actual components of the electron transport chain. And what they do is they act as proton pumps, right? H plus, that's just a proton. You're pumping protons from the mitochondrial matrix into the inner membrane space. And the energy from NADH's electrons is what is actually powering those pumps. What you end up getting is a very high concentration of protons in here. And remember, protons are what we're measuring in pH. So in case you ever see it pop up on an exam, high concentration of proteins also means low pH. So the inside of the inner membrane space is actually slightly acidic, right? The purpose of all this is these protons, because they're building up at a high level out here, they want to flow back this direction by diffusion. Now, the proton can't go straight through the membrane. It's too water soluble. That's not going to happen. But what we can do is we can take this gigantic enzyme called ATP synthase, and ATP synthase has a pore through the middle that lets those protons flow back through. And when protons flow through, that gives energy. So the actual gradient, the fact that proton levels are very high out here and very low in here, and they want to flow through, it's kind of like water flowing down a waterfall. If you put a water wheel in, you can drive a generator and make some electricity. This is the same deal. Protons flowing through ATP synthase give this enzyme the energy that it needs to take ADP and a free phosphate. This is that inorganic phosphate we saw earlier, and reattach them, right? It takes energy to do that. Phosphates don't just spontaneously reattach to ADP. And so we take the energy from our proton flow, use it to tack that third phosphate back on, make ATP, and that ATP is then going to go out to the cell and get used to release energy, right? And once you use it, you're going to take the ADP and phosphate right back into your mitochondria again and just reattach them. And so this entire process, that's all we're doing. We're taking our high energy electron carriers, right, NADH and FADH2, using their electrons to run the electron transport chain. End result, we pump all these protons into our inner membrane space. And since those protons want to flow down their concentration gradient back through ATP synthase, right, we use that energy to reattach a phosphate to ADP and make ATP. And so it seems like these protons are kind of just chasing their tail. And to some degree they are, but we're using them as an energy currency, right? That proton gradient is how you transfer the energy from NADH to eventually make ATP. All right. The one thing that I thought it would be helpful to tack onto this is a little bit more about the details of the reaction and where oxygen comes in. So this is a zoomed in view of the electron transport chain. Again, this is mitochondrial matrix. This is inner membrane space. This is where we're building up all of our protons. And here's that same ATP synthase that we saw before. I like this because it shows these little E minuses. This is NADH handing off its 2 electrons, and the electrons are jumping from one component of the chain to the next. Every time they jump from one electron transport chain protein to the other, their energy is going down, right? So these are very high energy electrons. By the time they come off the end, these are very low energy electrons. We don't want them anymore. So what do? They have to go somewhere. Electrons can't just float around by themselves. It's dangerous. They're very reactive. And so you'll hear oxygen called the final electron acceptor. And all that means is when the electron comes off of complex 4, the last part of the electron transport chain, they have to go somewhere. That somewhere is oxygen. So you combine one oxygen atom, right, half of an O2, because it's just one of the 2 oxygens in O2, plus 2 protons and 2 electrons. Remember, 1 proton plus 1 electron gives you a hydrogen atom. And so this half an O2 is where the oxygen comes from. And combining 2 protons and 2 electrons gives you 2 hydrogens, H2O. So when you see your big picture chemical reaction saying, right, glucose plus oxygen yields CO2 and water, The CO2 we lost back in Krebs cycle and pyruvate oxidation. This is where we're using our O2, and water is just the waste product. This water is just going to go into circulation, into cells, and eventually be eliminated from the body as a waste product. And so in the end, we get a lot of ATP here, right? The vast majority of your ATP is made at this step, and this is what we call oxidative phosphorylation, right? That is different than substrate level phosphorylation that you saw in glycolysis, because the phosphates in this case aren't being transferred from a substrate. They're just floating around by themselves and being directly reattached to ADP. Okay, so as one final overview, remember, assuming we're aerobic, we started with glucose entering glycolysis. We oxidized that glucose repeatedly to make pyruvate, in the process making a little bit of ATP, but also some NADH. If we had oxygen, we converted that pyruvate into acetyl-CoA, had it enter Krebs cycle, and in the process made a lot more NADH, right? A little bit of FADH2. And we eliminated the carbons as CO2 along the way that are going to go to your lungs and just be breathed out. All of these NADHs that we made, right, and the FADH2 are just electron carriers. So they picked up electrons in glycolysis and in Krebs cycle, and they handed those electrons off to the electron transport chain. We used the electron transport chain to harness the energy of those electrons and pump protons into the inner membrane space of your mitochondria. And then those protons flowed back through ATP synthase, giving it the energy that it needs to reattach ADP and a phosphate to make ATP. And now that ATP can leave your mitochondria, be used to release energy that's going to allow cells to do the work that they need to do. And then the ADP and phosphate that's left over are going to be returned back to mitochondria to get reattached again. And all the while, you have more glucose, or remember, fats or amino acids can also feed into these processes, coming in all the time to make sure the process keeps running and cells don't run out of ADP. All right. As always, I hope this was helpful. If you're interested in more detail, there will be more videos posted in the biochemistry playlist that I have. This is the end for the introductory bio version, but we'll eventually get to more details if you want to check out the biochem playlist to get a more advanced version of this. All right, as always, flag the playlist, subscribe to the channel. The videos always make more sense if you have them in context and can watch them in order.